So first of all, thank you very much for joining this webinar or seminar. And also big thanks to you two, Karen and Mike, for being such nice hosts and for really wanting to learn how you use the tissue facts and also the image analysis applications. And also, we are really proud to be part of the African Microscopy Initiative. And yeah, we think it's a great program to help scientists all over Africa and support them with their research. So this is really kind of a very good project. We're really happy to be part of it. But now to come back to TissueGnostics. So TissueGnostics is a supplier for tissue cytometers. We will come later on to what this exactly means. We are located in Austria, in the beautiful city Vienna. And yeah, I'm actually a product manager there. And yeah, so maybe we move on now to the more important stuff. So I want to give you a short insight about what you can do with what is tissue cytometry, what can you do with it, how can it help you with your research, and also I explicitly will talk about the system that you have here at AMI, because it's really a very, very nice system and you can do really a lot of really cool stuff. So my purpose here, or my intention today, is really to inspire you that you can really see what you can do with the system. So just a short introduction. So what is tissue cytometry? So you're all familiar with flow cytometry. So this is the phenotypic and functional analysis of cells in suspension, for instance, in blood or disassembled cells. But the problem with flow cytometry is that you basically lose the spatial context. So you lose the tissue context. And this is what we want to tackle with tissue cytometry. So tissue cytometry includes, first of all, the imaging of immunofluorescence or IHC processed tissue sections, TMAs, or cultured cells. And the second, and actually kind of more important part, is the image analysis. So you can detect molecules, specific phenotypes, you can detect morphological tissue entities, and you analyze all these things within the native tissue environment. So you do not destroy the tissue by this. So it gives you also kind of a spatial context. So the system that you have here at the AMI is the TissueFax SL+. So just a bit of an overview about the technical parts. So you can do fluorescence and bright field whole slide imaging. You can scan up to 120 slides, so it's really a high throughput system. You basically load all your slides, apply profiles, and then you just run it overnight, basically. So it's really it's a high throughput system. You have different kinds of objectives. With this system, you have a 2.5x. This is usually used for preview. So usually you first preview the slide, then you do some tissue detection, and then you acquire it in high resolution. And high resolution objectives, we have a 10x, 20x, and 40x objectives. Theoretically, if there's any need, it can be also upgraded. So we even have systems with 100x objectives. Yeah, so you can scan tissue sections on slides, also TMAs, fixed cells, but we will come to this later. You have different filter sets. I just added here the most common fluorochromes, but if you need more data on the filter sets, just go to Karen and Mike, so they have all the information. But in theory, we have 8 different filter sets. So you can export these digitized slides in a lot of different file formats. So you can export it as OME-TIFF, as a tissue or in TissueFax format, as JPEG, PNG. And this is important because we are a very open platform. So you can export it in any file format. And if you, do not have access, you do not have time to analyze your samples here, you can also analyze them theoretically with Fiji, with QuantStudio, or with any other image analysis software. Yeah, and then the last part, which is maybe the most important, is the analysis of the digitized slides. So we have 3 different softwares for image analysis. All of them are here available at AMI. Yeah, so what kind of samples can we scan and analyze with our TissueFax? So you can scan slides which have paraffin sections on them, cure cut sections, also biopsy materials, tissue microarrays. For tissue microarrays, since it's usually a lot of regions, so you can have up to 100 small tissue sections basically on one slide. It also has a specialized workflow for that. which is actually making the scanning of TMAs very easy. You can also scan cell smears, cytospins, chamber slides, and so on, but they should be processed with some kind of staining method. So either immunofluorescence or immunohistochemistry or all different kinds of morphological stains like H&E or trichome staining, etc. But now we come maybe to the more fancy part, which is the image analysis. So what can you analyze? You can, for instance, do just basic single cell analysis. You can do detection of multicellular structures. You can also do spatial phenotyping. You can also quantify cellular pathogens. You can do a detailed intercellular characterization, or actually anything else that you want to analyze that you can imagine. So our software is very powerful. So we can even, so usually when customers come to us and ask us, can we analyze this? Most of the time it's possible, even if it's not biological material. So we even have one customer who wants to detect holes in tires, yes. And even this is actually possible. Okay, but now I want to give you some more in detail examples of the capable of the image analysis. So we will start just with basic single cell analysis. So in this case, this is a colon sample stained for hematoxylin and for Ki67. So you all know Ki67, I guess. It's like the most known proliferation marker. And this is also maybe an example why image analysis is kind of important, because here we asked a lot of different experts how many cells or how much percent they think here in this image are positive for Ki67. So maybe you can also try to guess. I give you just a few seconds and you can think of a number. Yeah, just keep this number now in mind because you can see the expert estimations were from 1 to 40%. So you see already, it's very observer independent. Yeah, and with our image analysis software, we can basically just identify the nuclei, and then we can detect all the positive cells. So in green, you see now the nuclei detect with the negative nuclei, and in brown or in red, in this case, you see the Ki67 positive cells. And in the end, we get basically scattergrams. Everyone who is familiar with flow cytometry will know these kind of scattergrams. You can see here the Ki67 mean intensity. You usually set the cutoff based on an isotype match negative control. And then basically, you get a real number. So in this case, 15.49% of the cells in this picture were positive for Ki67. So now we move on to a different example. So maybe also what I should mention now is that all the samples that you see here were from customers or from our clients. So in this case, the client wanted to characterize tissue infiltrating lymphocytes. To be more precise, he wanted to detect T regulatory cells. So this is just a small tissue section, which was also again colon. It was stained for 4 markers and then acquired with tissue flux as well. So you have here TAPI in blue, CD4, which is a marker for T helper cells in yellow. You have CD8, a marker for cytotoxic T cells in red. And then you have also FOXP3, which is actually the marker for T regulatory cells. So usually when you detect T regulatory cells, you check for CD4, FOXP3 double positive cells. So what we did first was just the classical nuclear segmentation. And then, as I said, as output, you always get these kind of scattergrams. And now we come to the actually really cool part about the software. You see here the CD4 mean intensity. And all the cells which are above the cutoff, so which are now highlighted in orange, these are the CD4 positive cells. And then you can look basically into the image, select especially these cells, and they are then like shown in red in the image. So you can always go back to the image and verify your cutoff and verify also your results. And also it's working the other way around. So if I want to see where is this cell sitting in my scattergram, I basically just double click on it, and then it will show me where exactly it sits in all the scattergram. So here you can see now the detection of CD8 positive cells in the tissue. And here are our T regulatory cells, so the cells which are CD4 positive and FOXP3 positive. But as I said, so single cell analysis is just one part of the analysis, but you can also detect multicellular structures. So in this case, you see, I think it's pancreas tissue, and it's stained for endomycin to detect the blood vessels, and it's also stained for hematoxylin for the nuclei. And here the client wanted to detect, actually the blood vessels. So what you can do first, very easily, based on the brown channel, you can detect the endothelial cells. You can see now they are highlighted in brown, in green in this image. Then we detected next the blood vessels lumen. And in the end, basically, when you combine these two, it gives you the whole blood vessel. And again, by using the scattergrams, we could, for instance, gate by the size of blood vessels. So in the very right gate, you see there are the large blood vessels highlighted. So on the x-axis, we basically have the size of the blood vessels, and then we can basically highlight them again in the tissue. So you can see in red now all the big blood vessels. Here you can see the medium-sized blood vessels, and here you can see the microcapillaries. But you can also bring basically your blood vessels into spatial context if you want. So here it's again our pancreas tissue, and here we wanted to detect how many blood vessels are in the tumor and how many blood vessels are outside of the tumor. So first we just detected the tumors, and this was just based on nuclei density, because the tumors, just go back, they have a much more cellular density, a much higher cellular density than the rest of the tissue. Then the second step was to detect again the blood vessels, just based on the marker. And then in the end, basically, we can check how many blood vessels are inside the tumor and how many blood vessels are outside of the tumors. And you can then export, for instance, like in this case, the number of blood vessels, but you can also detect the area, you can detect the density of blood vessels. So there are hundreds of different things that you can analyze and export. But you can also do, for instance, some dot detection. So some, for instance, FISH analysis or RNA-scope or CISH analysis. Or also many people are using this dot detection algorithm for detection of viruses or other parasites. So here, for instance, is a FISH experiment. So again, we have the nucleus stained with DAPI. We have a housekeeping gene, which is stained in green. And then we have HER2 Neu in orange. And first step was we detected the nuclei. The second step was to detect the housekeeping gene dots. The third one was to detect the HER2 dots. And then in the end, you can see the combined mask. And then you can export basically the number of dots per cell. You can also export the total number of dots, so whatever you want to. And this is actually, so this algorithm is super easy to use. Yeah, so here you can see again basically the scattergrams, and what we did here was we just gated on cells which had 2 dots of each. So you can see now all cells which are highlighted here in red, they had they have 2 dots of the yellow one, which I don't know, I think it's the, I don't remember, maybe the acridine or the HER2 dots. So you can also very nicely gate again for specific parameters. Okay, what we also have now in our software, which makes actually life for researchers, especially when they do not have such a background in image analysis, much easier, is our machine and deep learning solutions. So we will come to that now. So this is an example also from a customer. So this is, I think it's a lymph node. It's for sure some kind of immune cell organ. And this was stained for 6 different markers, so for some immune cell markers. And you can see very nicely here the terminal centers. You can see in green the epithelium. You can see in pink the T cell zones. So it's actually very nicely stained. This was also scanned with our TissueFax, by the way, so it has 255 individual fields of view, which were very nicely stitched together so that you have this nice overview picture. Exactly, and you can see here just a zoom on the DAPI channel, and yeah, you can see that some areas are really dense. Especially, we have a lot of problems within such tissues with our classical nuclear segmentation algorithm. So what we developed is a deep learning-based nuclear segmentation algorithm, which tackles these kind of tissues much better. So this was the classical nuclear segmentation. And you can see already, so down there it's actually very nice, but the higher you move up, the worse is actually the segmentation. And this is our deep learning-based nuclear segmentation. So we miss some cells up there, but the cells that it detects actually perfectly. And this was the first version, so we are now at version 3, which will be also installed here at AMI. So you can try all these different deep learning methods as well. The thing is that sometimes the classical still works better. It just depends. So this is something you always have to figure out and try out. But for these immune cell organs, actually the deep learning one works very good. And maybe also to mention that when you use the classical nuclear segmentation algorithm, usually you have still to set some parameters. But with the deep learning one, it's basically just loading and running it. So it's much easier, I would say. Yeah, here's just again some comparison. And now we come to the second machine learning-based tool that we have now, and this is a classifier. So you can classify your tissue, so you basically substructures. So in this case, for this immune cell organ, we defined these different kinds of multicellular structures, or tissue architecture. Let me just think about a better word. Tissue substructures like germinal centers, mantle zone, T-cell zone, epithelium, exotonsillar space, muscle, soma, and endothelium. And in the classifier or in the software, you just mark or you just paint on the image, like here. See this red line. You just mark basically tissue areas according to the structures that you want to define. And then you train it based on these marked areas. And then in the end, you get a classification mask. Usually it's not 100% perfect, so you can still do some post-processing, but it gets already very good results within a short time. And then further, you can do some additional spatial analysis. So you can, for instance, check or look at all the cells which are detected in the germinal center. So they are now here highlighted in orange. And you can do a more in-depth analysis. So you can check how many cells are positive for the red marker, which I think was in this case a T-cell marker. You can check for instance, the blue marker, which was, I think, in this case, the B-cell marker. No, it was not a B-cell, maybe CD8 or something. And then you can also look at double positive cells, but only here in this case in the germinal centers or in any other structure. So now we actually come already to my last example, which is one of my favorite ones because it also gives you some proximity measurements. So in this case, the customer wanted to know where are all my CD4 positive T cells. So this is a colon cancer sample, and it was stained for 5 markers. So with DAPI in blue, the epithelium is stained in light blue. The CD4 positive cells are in red, so the T cells. We also have FOXP3 in green and KR67 in white. And what we did first basically was just a nuclear segmentation. You can see here also a zoom. Then we detected based on the epithelium staining, so this was a cytokeratin marker, we detected the tumor areas. Here you can see the combined masks. And here you can see a scattergram out of this analysis. And this, the y-axis is nuclei distance in micrometers. And the input gate here were only CD4 positive cells. So each dot that you can see here is a CD4 positive cell. And they are gated based on distances from the next epithelial area, so from the next tumor area. So all CD4 positive cells, which are in this gate, are 1 to 25 microns away from the next tumor area. Here we have 25 to 50, 50 to 100, 100 to 150, and so on. And it also gives us some quadrant statistics already, which you can see here on the right. So we have here, so it gives us exact numbers. So in total, we detected more than 7,000 T cells within the, or CD4 positive cells within the whole tissue section. And in a range from 1 to 25 microns next to the tumor area, there are exactly 2,067, and the number decreases actually the farther we go away from the tumor. So it already gives us some biological meaning. Yeah, and I can also visualize this in the tissue in the software. So in red, you see now all CD4 positive cells, which are 1 to 25 microns away from the next epithelial area. Here, 25 to 50, and here 50 to 100. Yeah, so actually, that's it with the examples. I hope I was able to show you a bit what you can do with it potentially. If you have any questions regarding image analysis, or if you have already any ideas what you could do with the system, yeah, just, I don't know, talk to Mike and Karen. Or to me now, when I'm still here. Yeah, and I also want to encourage you to look at our website. There is a section with reference publications, and we are trying to collect all the publications where TissueGnostics products were used for research. So there's also a keyword search. So if you want to search for a specific keyword to check if something was already done with our systems in your research field, yeah, please feel free to look through it. So we have an increasing number of publications, and we really try to update it monthly. Yeah, so that was it. So thank you very much. Yeah.